Let's move on by talking about cytoplasmic accumulations. These are forms of accumulation linked to four main causes: endogenous metabolic changes, faulty protein transport, enzyme defect regarding the enzymes metabolizing endogenous substances, and accumulations of exogenous substances. Depending on the type of accumulated biomolecule, there are different radical conditions. Let's talk about lipid accumulation, starting from triglycerides of fatty acids and steatosis. Steatosis may be linked to different causes, and the main one is the inhibition of beta oxidation. Beta oxidation is a reaction during which lipids are oxidized and converted into acetyl coenzyme with an enormous amount of ATP. Being an oxygen-dependent reaction, this inhibition may be linked to oxygen deficiency, which may be anoxic hypoxia with low oxygen pressure in the atmosphere, anemic hypoxia with reduced hemoglobin, and toxic hypoxia with molecules directly interfering with beta oxidation. Another example is that of the diphtheria toxin, which reduces the intracellular amount of carnitine. Carnitine then moves fatty acids into mitochondria, where beta oxidation occurs. Moreover, oxygen deficiency may be linked to the vitamin deficiency of the respiratory chain or to obstruction of bronchi, hindering correct oxygenation. In addition, blocking carnitine causes drug-related beta oxidation, and this phenomenon may be a side effect of two common drugs like zidovudin, an antiviral, and valproate, an antileptic drug. But steatosis may be linked to other nutritional alterations. During hypernutrition, increased fat intake leads to accumulation, but the same may be observed in case of fasting. This is because the peripheral tissue is activated and it metabolizes large quantities of triglycerides, thus increasing the amount of fat in the system. Similarly, in case of prolonged fasting, chronic protein caloric hyponutrition, such as Kwashiorkor syndrome, occurs, with consequent steatosis due to insufficient lipoproteins. Normally, to circulate, lipids need to be guided by specific proteins, the apoproteins. During chronic hyponutrition, the body doesn't have enough materials to produce these proteins, so lipids cannot move around and therefore accumulate. Finally, steatosis may be caused by toxins reducing the protein synthesis, such as ethionine, pyromycin, or carbon tetrachloride. Ethionine is capable of taking all the ATP, and pyromycin, together with tetracycline and alpha-amanitin, stop RNA polymerase. Carbon tetrachloride may cause different types of damage, but the main one is the endoplasmatic reticulum membrane peroxidation. One of the preferred sites of accumulation by triglycerides and fatty acids is undoubtedly the liver. The liver carries out a complex metabolic process of fatty acids, extracts them from the blood, then puts them together with the alpha-glycerophosphate, forming triglycerides. However, the liver can complex them with acetyl coenzyme A and form phospholipids or cholesterol esters. At the same time, the liver can also beta oxidize them, producing energy and, when necessary, ketone bodies. Moreover, triglycerides may fuse with apoproteins and then circulate as lipoproteins. Hepatic steatosis manifests as an accumulation of fat visible to the naked eye. The liver is 3 to 4 times bigger than normal and yellowish due to beta-carotene that accumulates in highly lipophilic tissues. From a microscopic point of view, we can observe two forms of accumulation: macrovesicular steatosis, where liver cells contain only one big lipid droplet caused by triglyceride accumulation, and microvesicular steatosis, where accumulation is fragmented and caused by fatty acids. Being usually characterized by a general accumulation throughout the cytoplasm, microvesicular steatosis is a more severe form. Lipid accumulation is positive to SUDN4 but are PAS negative. There are different forms of hepatic steatosis, but the most important is alcohol-induced. Alcoholic fatty liver disease is related to ethanol or ethyl alcohol consumption for food or recreational purposes in many countries. The metabolic process in this case is quite complex, and the liver converts ethanol or ethyl alcohol into acetaldehyde. This conversion may be carried out by catalase in peroxisomes or by alcohol dehydrogenase. Alcohol dehydrogenase is an enzyme that dehydrogenates alcohol by moving a hydrogen on NAD+ and converting it into NADH. This reaction may occur in the smooth endoplasmatic reticulum of the microsomal oxidation system, a system based on P450 cytochrome that oxidizes ethanol thanks to an atom of iron. This is an inducible system because P450 cytochrome effectiveness may be increased. In fact, the more P450 cytochrome works, the more its power increases due to the higher number of transcribed cytochrome enzymes when cytochrome is under stress. However, P450 cytochrome doesn't catabolize ethanol only, but a large number of drugs. Therefore, a chronic alcoholic has an extremely active P450 cytochrome, rapidly catabolizing most of the drugs taken and therefore with lesser effects. Acetaldehyde may end up in different ways, but must be eliminated due to its frequent conversion into a radical form. One of the simplest metabolic forms is that of dehydrogenase acetaldehyde that converts it into acetate, producing NADH. The reason behind steatosis formation caused by ethanol is that the DNH produced in these phases is used for lipid synthesis. Ethanol indirectly increases lipid synthesis. Acetaldehyde may be either catabolized by xanthine oxidase that converts it into carbonate radical, or, if in a radical form, may carry out lipoperoxidation of that in mitochondria or endoplasmatic reticulum, leading to protein synthesis and cellular respiration interruption. Moreover, acetaldehyde may be metabolized by glutathione, leading to the reduction of the active amount inside the cell, and finally may form attacks with the proteins that stimulate a chronic form of inflammation, resulting in fibrosis and therefore in cirrhosis. However, hepatic steatosis may not necessarily be linked to alcohol. If so, it is linked to the dysmetabolic syndrome. During insulin resistance, we can observe a deregulation of normal lipid metabolism. For example, saturation of beta-oxidation systems with consequent lipid accumulation and no alcohol consumption is quite common in obese people with this metabolic syndrome, eating too many fats. Another non-alcohol-induced cause is that of several drugs as amiodarone, corticosteroids, and tamoxifen that directly interact with our metabolism. Finally, pregnancy interrupts lipid metabolism by reducing the amount of 3-hydroxycyl-CoA reductase dehydrogenase, an enzyme of the beta-oxidative cascade that stops lipid catabolism. The heart is another organ where lipid accumulation may occur. From a macroscopic point of view, since it looks striated, the heart is called cor trigatum, from Latin. From a microscopic point of view instead, lipid accumulation among muscle cells, apparently due to hypoxia, may be observed. In case of moderate hypoxia, the Steiger skin appearance is more evident, while in case of severe hypoxia, the heart is almost entirely yellowish. Cholesterol and cholesterol ester accumulation is another example. Lipid accumulation may be also genetic. There are a series of pathologies called familial hypercholesterolemia that are caused by metabolic alterations of LDL receptor. This receptor normally takes LDL from the blood and puts them mainly into the liver. If there is no receptor, a large amount of cholesterol circulates. Type C Niemann-Pick disease is another genetic form where NPC transporter, an ATP binding cassette, whose function is to carry lipids intracellularly, is altered. This phenomenon results in large accumulation. Acquired causes of accumulation are a lot more common. Diabetes mellitus, for example, blocks beta-oxidation. Hypothyroidism slows basal metabolism and therefore beta-oxidation. Hepatitis blocks biliary excretion, which is one of the simplest ways for the body to release cholesterol and cholesterol esters. Nephrotic syndrome causes cholesterol accumulation in tissue through urinary loss of lipoproteins. Finally, the use of corticosteroids are a very bothersome metabolic effect, which is the increase of fatty acids to circulate again and accumulate in tissues. From a morphological point of view, cholesterol accumulation may be in different forms. The most fearsome is atherosclerosis, a phenomenon where damaged lipoproteins insert under the enema of the large vessels damaged by shear stress, and then triggering significant inflammation involving macrophages. Lipoproteins are phagocytized but not catabolized by macrophages that engulf and then release cytokines, worsening the inflammation. Then cholesterol plaque made up of dead macrophages and active macrophages is formed. By breaking, this plaque leads to its inner lipid core exposure and therefore coagulation. This mechanism is extremely common in myocardial infarctions. A less severe form is that oxanthemas, voluminous mass of foam cells, normally engulfs macrophages that accumulate subcutaneously and in tendons. Finally, cholesterolosis is an accumulation of foam macrophages in the gallbladder lamina propria. Thank you for watching. This video was created by School of Biomedical Sciences, Agora. We hope you enjoyed it. If you're curious or have any doubt or question, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the following link to visit our website.